when we will have a symposium, GSI Research Symposium, which you should look for information about that. And so today, our guests who are presenting seminar, Alyssa Nolan from 10x Genomics and the team. And 10x Genomics is a great partner of BU and BU's sequencing facility. They supported a lot of our workshop initiatives by providing reagents, and we really appreciate it. And the topic of this seminar was actually named the Method of the Year by Nature Methods, and it's a spatially resolved transcriptomics. And it's 10x Genomics is on the cutting edge of methodology for sequencing. They are standard in industry for single cell RNA-seq, and now they will present us the new platform Visium for spatial resolved transcriptomics. Alyssa, please go ahead.
Thank you, Alla. That was a great introduction. Hi, everyone. I see some names that I recognize, and it's certainly nice to virtually meet you. I'm so glad that we're at least able to host or participate in virtual seminars, even if we can't do anything in person at the moment. But as Alla mentioned, we're so excited to be able to talk today about our spatial transcriptomic solution, which we call Visium. And Visium in the past had worked with just fresh frozen tissue, and now we have an additional product where we can access that same transcriptomic information from both from FFPE preserved tissues, which is really exciting. So we're going to focus a lot today on the type of data that you can generate. We're also going to focus on some more practical and technical based tips and tricks on how you might get this to work in your lab and in your hands. So hopefully you'll find that useful. Just a quick intro to the team and myself, certainly. So we're all local. We're all based in the Massachusetts area. I am the sales executive that supports the researchers at BU. I'm joined today by my fantastic colleagues. We have our tissue specialist, Shoni Draws, who is joining us. She is a great resource for all things related to tissue sectioning and placement and preservation. So she is certainly a resource I want to make sure everybody is aware of. And then we have our two presenters today. So Rosha Padyal is our local science and technology advisor. And with her is Stephen Wood, our local FAS field application scientist, both of them supporting the BU community. So certainly feel free to type questions in the chat, ask questions as we go. We should have enough time to be able to address some questions at the end as well. So if you want to wait until then, feel free. And certainly we're available after the seminar as well if you want to have further discussion. So without further ado, I will turn this over to Rosha and Stephen.
Thanks, Alyssa. It's nice to see everybody. Thank you for the great introduction. Thank you, Alla. Thank you, Alyssa. Thank you for having us today. I was actually just thinking about the last time we 10x presented at one of these meetings, and I think it was right before, it was March of 2020, and I had come on site to talk about some of our single cell applications. It's really nice to be back again, to be able to talk to you about a completely different solution, which is the Visium spatial transcriptomics. So what I'd like to do in the first 10 or 15 minutes today is give you a little bit of an idea about how the technology itself works, the basics of the technology. And Visium for FFP was recently released about a month and a half, 2 months ago. So we do have some internal data and how we've utilized this technology to answer some biological questions. I'll walk you through that. And then I'll pass it on to my fantastic field application colleague, Stephen, who's going to talk through the details of how the applications of Visium and the workflow overview and the technical tips and tricks. So before we get started on the details of the technology itself, there's a lot of buzz about spatial biology, and just a basic question that we might ask ourselves is, what is the significance of spatial biology? So we can look at a tissue section. Of course, pathologists who are used to looking at tissue sections can then, for example, when we're looking at something like the human breast ductal carcinoma in situ, they can look at and annotate which regions might be tumor, which regions are stromal, what kind of activities might be going on there. But what Visium lets us do is it lets us overlay this unbiased whole transcriptome data on top of the tissue. So we can now start revealing these complex molecular environments and start investigating the molecular underpinnings of these diseases. A lot of times we will see molecular changes before we can see phenotypic changes or pathological changes. So when you have this unbiased molecular transcriptomic data, you can start seeing different clusters and digging deeper into this data set to understand this biology furthermore. And like Ala mentioned, spatial biology was termed the Nature Method of the Year in 2020. And like Alyssa mentioned, when Visium was first launched, we supported fresh frozen tissue. So even with the fresh frozen tissue, there's been a lot of publications that have come out that utilize Visium in neuroscience, oncology, developmental biology, immunology, really anything where you can think the tissue context is going to be beneficial to either understand what kind of cells are interacting with what's going on in the tissue environment. Visium can be applicable to all those areas. And so recently we did launch our Visium for FFPE solution. This, of course, again, captures the whole transcriptome, and this is a probe-based method. And I'll walk you a little bit through how exactly that works. Being a probe-based method, it means it has, the probes are designed currently for human and mouse genomes. We target about 18,000 genes in the human genome and about 20,000 in the mouse genome. There's a lot of mitochondrial and ribo genes that are depleted that the probe sets don't have. So that's why the number is a little bit lower. But here you can see an example of when you have these FFPE tissue sections that usually shrink, the molecule, the RNA molecules degrade, you don't get great data. If you were trying to use a poly-A approach, like you would with fresh frozen tissue, but you can get pretty nice clustering data sets with both the human and mouse probe sets for Visium for FFPE. So Stephen will walk through the details of each of these steps in part of his presentation, but the overall idea of the spatial gene expression Visium for FFPE is that you start with a piece of tissue that you're interested in, and you section it and you layer it on what we call our gene expression slide. So you can see there are 4 different, each of these is a single capture area. So we have 4 different capture areas where you layer on your tissue slide. That is followed by, if you're interested in morphological context, you'll do H&E staining. Or if you're interested in immunofluorescence, you'll do protein code, or if you're interested in protein code detection, you will do immunofluorescence. And of course, for FFP, there's crosslinking, et cetera, that Stephen will also touch on. So this is just basic overview. And once you have your tissue imaged, then you can perform barcoding and library construction. Part of it happens on the slide, rest of it happens in a tube. And what you've generated at the end of that workflow is a sequencing library that you can then sequence on an Illumina instrument. And we do have data analysis pipelines as well as data visualization tool that's available to you at no charge to analyze your Visium for FFPE dataset. So the way these probe sets work, this is RNA templated ligation. So what we've done is we've designed these probe pairs against the protein coding transcriptome, and there's 1 pair per gene. So we like to call this the left-hand side probe, and you'll see this has a 5' read 2S sequence on the left side. And we have the right-hand side probe, which has a poly-A tail. So this is a synthetic poly-A tail. Once you have your mRNA target site, these are of course going to be degraded, and our algorithm has come up with this probe sets that work pretty well. What happens is these probe sets will now find the target and ligate to each other. So these are pretty specific because you have to make sure both the left and the right-hand side probe pairs find each other. And once it finds the target mRNA, what we actually do is we digest await the original RNA molecule. At this point, you'll notice we have added this synthetic poly-A tail. So what that means is on a gene expression slide, there's this oligonucleotide that has poly-DT, and using this poly-A tail, now we can capture this information here. So we're capturing these hybridized probes as a proxy of what the original target mRNA was present in the tissue slide. So if we looked a little bit closer into the construction of what's laid down on a capture area in a gene expression slide, it looks something like this. So the capture area itself is 6.5 millimeters by 6.5 millimeters. The outside edges is what we call our feudicials. There are 4 different colors, 4 different shapes on the 4 different corners, you'll notice. And that's important because when you align your transcriptomic data with the image data, the software uses these as kind of a guide to align it properly. So the entire capture area is 8 millimeters by 8 millimeters, but the inside part where you have these spots where you will be generating the transcriptomic data that has oligonucleotides is about 6.5 by 6.5 millimeters. If we zoomed into the capture areas a little bit more to see what kind of spatial barcodes are available. So there are about 5,000 unique barcode spots. And if you zoomed into any of these barcoded spots, it's a lawn of oligos. So there is hundreds of thousands, millions of oligos, where the construction of oligo is something like this, where we have this partial read one, it's a PCR handle, sequencing primer. Then we have spatial barcodes. So each of these spots are going to have a unique spatial barcode. And we have a UMI to so there's some amplification steps. We want to make sure we get rid of PCR duplicates, so we utilize the UMI, and we have the poly DT. So of course, we're capturing the synthetic poly A tails on those probes using this poly DT. So when you sequence this data set and you figure out which particular spatial barcode is corresponding to a particular transcript, then we can overlay that molecular information onto this capture area that also will have the image information. So that's how you're getting these 2 layers of information. The output, of course, is going to be an Illumina sequencer compatible library. So a sequencing depth, usually the questions come up about how deep should we sequence, and we recommend sequencing about 25,000 read pairs per spot for one of these experiments, but that can be a little bit for example, if you're working with something very small, like an embryonic mouse, mouse brain that probably didn't cover all of the capture area, then of course you wouldn't sequence 5,000 spots. You would do the proper multiplication to figure out what your sequencing depth might be. So as I mentioned, we released this product recently. We've seen some customer data in webinars where they've utilized Visium for FFPE, and it looks pretty good, but I wanted to quickly walk you through something we've done internally, and we presented this data at EGBT as a poster. And this is a little bit of just exploratory work we did with triple positive ductal carcinoma in situ of the breast. So triple positive breast cancer, it's rare. 10% of all new breast cancer diagnoses each year is TPBC, but it is pretty aggressive. And the major signature for a TPBC is, of course, there's positive for cell surface proteins and genes of HER2, estrogen receptor, and progesterone receptor. So the goal really here was to take this tissue and try and understand the TPBC heterogeneity and profile the tumor microenvironment using Visium for FFPE and see what we would be able to discover. And this was done in a little bit different way as well, because we also wanted to make comparisons with our fresh frozen product. So what we did was when we had access to this tumor, what we're able to do is use 1/3 of it for FFPE embedding, the other 1/3 for fresh frozen embedding. And we actually used 1/3 of the tissue to isolate into single nuclei and perform a single nuclei RNA-seq experiment with that as well. So some of the comparison between fresh frozen and FFPE data sets, for example, in the middle panel that we're looking at here, we're looking at the correlation between the data set for FFPE versus whole transcriptome. The correlation is pretty good. Of course, it's not 100% because we're not looking at the exact same section, because by virtue of what we were trying to do, some of it had to go through FFPE, some of it through fresh frozen, but the correlation in general looks pretty good for the data set. And we also looked at multiple sections of the FFPE tissue to see what kind of correlation we would get between serial sections. And that looks pretty good for a Visium for FFPE as well. So some preliminary work, of course, is to look at the pathologist's annotation and see how that aligns with the whole transcriptome cluster data that was generated using our software. And so on the left, we have the pathologist annotation here. I'll bring your attention to the regions that are shown in yellow. So that's the ductal carcinoma in situ. And we've similarly also have the red clusters here that are showing that are shown on the using the whole transcriptome analysis also is showing what ductal carcinoma in situ would be. So now you can dig deeper into this particular cluster and start looking at the molecular changes there. The other affirmation experiment that or data to look at is, of course, how does the immunohistochemistry for the 3 known protein markers correlate to what we see from Visium. So in the bottom panel, we have immunohistochemistry for progesterone, HER2, and estrogen receptors. And on the top panel, we have the gene expression for the corresponding genes. And the correlation looks pretty good. So again, where we see this expression of those particular genes is where you also see the proteins. But the cool thing here is because this is the whole transcriptome, you're not really limited to looking at particular sections. So what we can do is, for example, we know T-cell infiltration is in the tumor microenvironment is a hallmark of the immune response in cancer, and it can have a very good prognostic value in the clinic. So because this is an unbiased whole transcriptome data and we're not restricted to looking at particular markers, now we can start looking at, for example, CD4 as a T-cell marker to see where CD4 is infiltrating in this particular tissue. We can look at macrophage markers and any other kind of markers you're interested in as well. So a lot of that is validation and things that are known, but we also try to touch on something that would be novel and something that a lot of people are now interested in, which is looking at B cells. So recently, the importance of B cells in the tumor microenvironment is being investigated a lot more and being discussed a lot more. And the role of B cells in the tumor microenvironment seems to be pretty diverse. So besides secreting antibodies and cytokines, the B cells are also hypothesized that they are able to modulate the T cell and innate immune response as well as recognize antigens and regulate the antigen processing and presentation in the tumor microenvironment. And what's been particularly shown at this point is that there is a balance between the B cell subtypes. And the subtypes, of course, are characterized by the expression of specific markers, and the activities can be pro or anti-tumorigenic function of these B cells, depending on which subtype is being expressed in the tumor. So B cells go from, you have memory cells that turn into plasmablasts, and you have plasma cells that are also found in these tumor microenvironments. So when we looked at the single nuclei data, what we're able to find is, of course, we have the plasma cells, we have plasmablasts, we have a population of memory B cells, and using some computational tools, we're able to corroborate these data sets and collate these data sets together to see different stages of B cell maturation and where these cells were found in that particular tissue. So for example, if we're looking at memory B cells, we can see some expression up here, higher expression where it's more red. And if we're looking at plasmablasts, they're in a different location. If we're looking for plasma cells, they seem to be a lot more spread out within the tumor. So this is just starting to scratch the surface of what's possible to do with a single experiment and all the depth of data that we have. And we can start investigating these data sets in a lot more depth and try to come up with newer hypotheses about the importance of B cells in the tumor microenvironment. So with that, I hope you gave you a quick primer of what the Visium for FFPE solution is and some of the very exciting work you can do with this solution. And at this point, I will pass it on to my colleague, Stephen, who's going to walk us through the technical details of the Visium for FFPE workflow.
All right, thanks so much, Rosha. Let me go ahead and get my screen shared here. All right, so everyone can see my screen and hear me? Great. So my name is Stephen Wood, and I'm the field application scientist that covers a large area of the Boston area, as well as the BU campus. And so today what I'm going to do is present on our Visium spatial gene expression for FFPE solution and walk you through an overview of how this protocol is going to be carried out. So again, just to give you a bit of context here, we have our 3' and 5' single cell solutions that you might be aware of. And what this allows you to do is to profile the gene expression profiles of the samples that you're going to be looking at through those assays. With our Visium solution, we can do a very similar thing, but in addition to getting the gene expression profile of those tissue samples, we are also now able to look at the spatial context of those samples to see what cells are being expressed in different locations of the tissues that we're evaluating. So when we first released our Visium solution, the samples that we were working with were snap frozen and OCT embedded. And this allowed us to directly capture the mRNAs that are being expressed in those tissue samples. But we're well aware that there is a large collection of formalin-fixed paraffin-embedded samples in biobanks and just in research labs that are frequently used because they are excellent at preserving tissue morphology and they're great for long-term storage. Unfortunately, these FFPE solutions, or these FFPE blocks are not really great for the Visium solution in its present form because that formalin fixation is going to result in cross-linking of our nucleic acids to proteins, and it's also going to introduce degradation of some of those nucleic acids. So it makes it really challenging to actually profile the transcripts in those samples. So in order to address that and unlock all of these FFPE blocks that might be available and just sort of sitting to be evaluated, we developed our new FFPE solution for Visium. So when you try to compare our current Visium solution, you can see our gene sensitivity that we can see with fresh frozen tissue. If we try to apply that to FFPE tissue, we have very, very poor sensitivity. We just are not able to profile the samples because of that degradation, because of that cross-linking that's occurring to those transcripts. But with the probe-based approach that we're taking with the Visium for FFPE, you can see that we're now able to recover a tremendous amount of that sensitivity, and we can really now profile these samples that are FFPE tissue blocks. So now we still have our standard Visium solution available, and that allows you to do that direct capture of our transcripts from those tissues. But now we can utilize our Visium for FFPE solution that allows us to indirectly target and identify those transcripts in these FFPE tissues, and then we can profile them just like we were doing before with Visium. We are able to do this with both human and mouse samples, and we've tested this across a wide range of different tissue types. And so these are just some examples that you can see. So it really is very flexible, and thus far has seemed to work across a very large number of different tissue inputs that have been tried out. Big picture, the way this workflow is going to work is we're first going to begin with an assessment of our tissue blocks. So we need to do an initial quality assessment to make sure that these samples are not too far degraded because we need to have a minimum quality there in order to successfully generate our transcriptional libraries. Once we've done that assessment, we're going to section our samples onto our Visium gene expression slide, and then we can go through first H&E staining, and then we're going to stain that and image that. Additionally, the assay is compatible with doing immunofluorescence. So if you don't want to do H&E imaging and you want to do immunofluorescence on those samples, we do have a protocol that is going to accomplish that as well. I'm not going to cover that in today's talk just because we're constrained for time, but that is a possibility for this assay. Once we've gone through the H&E staining and imaging, we're going to then go into the library preparation steps. So we need to decrosslink our samples and then go into that probe-based assay. We're going to hybridize our probes, capture them onto our gene expression slide, and then create our final sequencing library. Those final libraries can be sequenced in any Illumina sequencer, and then using our Space Ranger pipeline as well as our Loop browser, you can analyze and visualize the data sets. That's something that we're going to have to save for another time. I'm happy to have a conversation about those steps, but because that's a much larger topic as well, we just don't have the time to cover that aspect of the workflow. So Rosha had shown a similar slide to this, but I just want to reiterate that on the gene expression slide for Visium, we have 4 separate capture areas. So for the FFPE solution, we can use that for 4 serial sections, or you can look at different samples on that single slide. Each one of these capture areas, the fiducial area is about 8.5 millimeters square, but the capture area that has our spatial barcoded spots is going to be 6.5 by 6.5 millimeters. And there's about 5,000 spots within each one of those capture areas. So the first thing that we need to do is prepare our samples. And we have a number of very helpful guides available on our support webpage, which are going to walk through all of these different steps. So this tissue preparation guide is going to go through tips and best practices of handling different samples, working with FFPE tissue blocks, sectioning those samples, and then getting them placed onto that gene expression slide. I'm not going to be able to go through all the details that are contained in this guide, but that's something that you can read through this, and we would be more than happy to have a follow-up conversation to walk through some of the specifics of placing the sections on the slide. But in general, what we're going to do is take our tissue block, and we first need to expose that tissue. So if you have an FFPE sample block, there's going to be some paraffin wax over top of that tissue. So we first are going to take some thicker sections to remove the excess wax until we can expose our tissue using the microtome. Once we've exposed our tissue, what we're going to do is just take a couple of those sections, and we're going to process them to extract the RNA from those samples. And we're going to evaluate the quality of that RNA to see if it meets a minimum threshold to carry forward into the assay. And if it doesn't, we're going to have to use another block that will meet that threshold. Once we've identified the blocks that are going to be compatible for the assay, we can go ahead and section those and then place them onto our slide. So the quality assessment that we're going to perform is identifying the DB200 score for these samples. So as I mentioned, when we do the formalin fixation, that's going to result in some degradation of the nucleic acids in here. And so we want to see how degraded these samples are. We are going to target a DB200 score greater than 50%. And the DB200 score is simply a measure of the percentage of nucleic acids that are greater than 200 nucleotides. So we're going to take those samples, we'll process them just using like an RNA-easy kit, and then run that out on a bioanalyzer, and then look for those samples that have a DB200 score greater than 50%. And if we're able to meet that, then we have greater confidence that we're going to successfully generate libraries for sequencing. So what I'm showing in this slide is all of the major steps, and we're going to walk through most of these together. So we've identified now our tissue blocks that are going to be successful, that they have sufficient DB200 scores. We're going to section those and place them onto a slide. Then we're going to carry out some drying steps and desiccation steps, which are going to help adhere those tissue sections onto our slide. Then we're going to go into our H&E staining protocol. So here we're going to first de-paraffinize our samples. Then we're going to go into H&E staining, imaging those samples, and then we can de-crosslink and hybridize our probes. After we have hybridized our probes, we're going to carry out all of these additional steps where we're going to generate our final sequencing libraries that we can then sequence on an Illumina sequencer. So keep in mind, the capture areas that we're going to place our tissue sections onto is 6.5 millimeters by 6.5 millimeters square. So we need to get our tissue into a size that fits within that capture area. So after we have faced our FFPE blocks, what we need to do is, using a razor blade, cut away any excess wax that might occlude the fiducial frame that's going to be surrounding that capture area. Additionally, if our tissue is greater than that 6.5 square capture area, what we're going to have to do is score that tissue either in half or into 4s so that we can capture that tissue onto multiple capture areas. I'm not going to go through the specifics of that. Again, that's available in our guide. The other thing that I will point out is on our website, we have a number of really helpful videos that are going to walk through all of those steps. So that would be a really great resource that you have. Also, we're very happy to have conversations if you have any questions about that. Well, we're going to go ahead once we have trimmed our blocks and then sectioned our samples. What we have to do is float that onto a warm water bath, and then we're going to capture those sections onto our gene expression slide. Once we've done that step, we need to dry those samples using either a slide dryer, or you can also use a thermocycler adapter that we provide as part of the accessory kit for the Visium for FFPE solution. And that is going to fit onto a thermocycler and act as a drying surface. Or if you have a hot plate that is able to maintain constant temperature, you can utilize that as well. So we're going to dry our samples for 3 hours at 42 C. And this is, again, just going to help promote that tissue adhesion onto our slide. After that step, we're going to go on and desiccate these samples. So we're going to place it into a desiccator for at least 12 hours. And this is also a pause area that you can stop the assay at this point and hold it in the desiccator for up to 2 weeks before proceeding on with the rest of the assay. Keep in mind just sort of best practices for getting our tissues in the proper location. Ideally, what we want to have is all 4 of our tissue sections nicely laid out in those capture areas. We don't want to have excess paraffin wax overlaying other tissues that might be below or above other capture areas. We want to make sure that our samples are effectively dried. We don't want to see any kind of tears or folding in that tissue. Otherwise, we're not going to get great images out of those samples. And that's also going to impact the gene expression data that we get. We want to make sure that those samples are also within that capture area. So if you have tissue that's too large, those samples are going to need to be scored so that they're separated into different capture areas. The placement of these tissues onto that gene expression slide is a challenge, even if you're someone that's familiar with histology and is well practiced with placing FFPE tissue onto slides. And the reason for that is we're targeting a really specific small capture area. And so for that reason, we really strongly recommend that you take a lot of time to practice placing tissues into those capture areas. So what you can do is go into that tissue, our tissue preparation guide. And at the last page of that guide, we have a little template that you can print out, and then you can take some standard microscopy slides and trace over this little cutout. And what we have when you print that out on an 8.5 by 11 page, it's going to meet the same specs as a microscope slide, and you can just trace over with a marker those capture areas, and then you can use that to practice placing any other tissue that you have available onto those capture areas, because that's going to take some real effort and practice to get it precisely into those capture areas. So once we have our slide prepared, we're going to go into the H&E staining protocol. So I'm going to cover now just the H&E staining. Keep in mind, we do have a separate protocol that's going to cover immunofluorescence. Some of the steps are very similar, but some of the steps also get switched around just because of the nature of working with FFPE samples if you're trying to do immunofluorescence. So this demonstrator protocol is going to walk through all of the steps that we need to appropriately stain our samples, image them, and get them ready for the rest of the assay. So the major steps that we're going to carry out here, first is De-Paraffinization. Then we're going to go and do the actual H&E staining. We'll cover slip these samples using just an 85% glycerol solution. We'll cover slip them, and that's going to aid with getting better images out of them. And then we'll remove that cover slip and carry on to our de-crosslinking step, getting ready for that probe hybridization. So the first step is to do the de-paraffinization to remove that paraffin wax. The assay is going to require using xylene to remove that paraffin wax. Unfortunately, we have not identified an appropriate substitute for xylene. So just be aware that xylene is a really nasty chemical. It really requires you to work in a hood. So please contact your EHS department to see whatever regulations are necessary for handling xylene in your lab. So the steps for the de-paraffinization are shown here. We're first going to incubate our slide at 60 degrees for 60 C for 2 hours. So we can either use an oven or a thermal cycler. And what this is doing is just helping to melt and loosen that paraffin wax. And then we're going to do 2 rounds of immersion into xylene. And then we're going to wash that xylene out using ethanol. We'll then carry on to place that on a thermal cycler, again, just to remove any of that excess ethanol. And then we go through sequential washes with first 100% ethanol, then 96%, 70%, and then finally into our milliQ water. At this step, we're going to go directly into staining with H&E. So we're first going to add on 1 ml of hematoxylin stain to our sample. So we're going to just apply that directly over top of the slide. We'll remove any excess after that 3-minute incubation with the hematoxylin. We'll wash that in milliQ water. And then we're going to apply our bluing buffer for 1 minute. We wash that again in milliQ water, and then we're going to apply our alcoholic eosin. Keep in mind, just if you have previously used Arbisium for fresh frozen solution, the hematoxylins and each eosins that we're going to be utilizing are different for the FFPE. So we're going to utilize an alcoholic eosin for the FFPE samples as opposed to the one that is used for the fresh frozen. After our eosin, we'll wash that, and then we are going to proceed with cover slipping that sample and imaging that on our microscope. So we do have another tech note that's going to walk through all of the guidelines for imaging. I'm not going to go through much of this, but just be aware, we have validated some imaging systems. Obviously, though, there is a whole host of different microscopes and imaging systems on the market, and we can't validate every single one of them. For the FFPE solution, we do need to have a color camera in order to image those H&E images. You also are going to want to have a motorized stage so that you can image multiple capture areas at high resolution and then stitch together that mosaic. For FFPE, we only need a bright field color camera. If you were doing immunofluorescence, obviously you would then also need to have a monochrome camera that's able to capture those immunofluorescence images. The next step that we're going to do is carry out our tissue cross-linking. So remember, when we formalin fix these samples, we have cross-linked our nucleic acids with the proteins in our tissue. And if we were just to proceed with the assay at this point, we would have very poor detection of our transcripts because they're going to be basically hidden by that cross-linking. So we need to go through a de-cross-linking step so that we can separate out those proteins and those nucleic acids so that we can come in with our probes to detect our transcripts. So this is typically carried out either through doing a proteolytic approach where we would apply an enzyme to chew up some of those proteins. We can also utilize a heat approach. And so that's what we're going to use in our assay. In order to do this, we're now going to move to taking that slide and carrying out some of the chemistry on that slide. And in order to do that, what we're going to do is take that gene expression slide that has our tissue that we've used for H&E staining, and we're going to apply it onto this cassette assembly that comes as a part of the kit. And there's this rubber gasket that is going to essentially generate 4 wells that we can use to directly work with the tissue on that slide. And so we just place it into this cassette adapter, and now we have 4 wells that we can work with. So we're going to do that for the following steps for the de-crosslinking. And we're first going to begin by applying a 0.1 normal HCl solution to our samples. And so these are going to de-stain them from that H&E, and that helps to recover some of the transcripts in our sample. So we'll do that 2 times, and then we're going to neutralize it by placing it in a TE pH9 buffer. And that TE buffer is also going to help with that de-crosslinking step. So after we have that in the TE buffer, we're going to apply a slide seal, so just a clear slide seal that you would use for like a qPCR plate. We'll apply that over top and then incubate our sample at 70 C for 1 hour. And this is going to carry out that de-crosslinking. So at the end of this step, we can now go into our final step of the assay, which is actually doing our probe-based approach to capture those transcripts. So here we have available either a mouse probe set or a human probe set. And so depending on the nature of the tissue that we're going to be working with, we'll use the appropriate pair of probes. We're going to carry out a hybridization and then a ligation of those probes. And then we're going to capture those probes directly onto our slide. So Roche kind of covered this in her presentation, but I just want to reiterate that what we are doing here is taking 2 probes for our specific target site. So we have 1 probe set per gene, and these are going to be composed of a left-hand side and a right-hand side probe that is going to be directly targeting an adjacent area of our mRNA target. On the left-hand side probe, we're going to have this read2S sequence that's going to act as a PCR handle. And then on the right-hand side probe, we're going to have a synthetic poly-A tail that we're going to use to actually capture these probes onto our Visium slide. So we're going to carry out that probe hybridization that's going to hopefully allow our probe sets to both hybridize right next to one another on our target mRNA. So the way we're going to carry that out is using our slide cassette. We're going to add in our probes, and then we're going to carry out a probe hybridization for 16 hours to 24 hours to carry out that hybridization. So we can either utilize a oven that we can heat to 50 degrees, or we can just simply use a thermocycler and hold it at that temperature to carry out the hybridization. The next step is to actually ligate these probes. And the reason why we're doing this templated ligation is that this increases specificity. So if we had fragmentation and we didn't get both of these fragments actually ligated together, we're either not going to capture it with our polyadenylated transcript, or if the left-hand side was missing, we wouldn't be able to amplify this in our library, and so it wouldn't be showing up in our data. So by doing this left and right-hand side approach and then carrying out this probe ligation, we really increase the specificity of this assay. So we're going to carry out that ligation, and then we're going to go on and remove any of that RNA so we can just capture those probes. So we're going to digest our RNA in our sample just using an RNase, and then we'll proceed with permeabilizing that tissue to capture that onto our slide. So we'll do this in an extended run on our thermocycler. We're first going to pre-equilibrate our thermocycler adapter at 37 degrees. We'll hold it at that position. And then we're going to carry out that RNA digestion. So we do that for 30 degrees, applying our RNase to that sample. Then we're going to wash our sample and then apply our permeabilization enzyme to permeabilize our tissue and allow those probes to be captured by our gene expression slide on the captured area. So once we have capture, we have permeabilization and then capture of our probe set, the poly-DT capture oligos that are on the surface of our slide are going to capture that synthetic poly-A tail on the right-hand side of our probe. And then we're going to carry out a probe extension of our probes. and that is then going to be denatured so that we can pull off those probes from the slide and carry out the rest of the steps just into a PCR microstrip tube. So what we'll do is wash our sample 2 times with FSC buffer, and then we're going to denature our probe set with a 0.08 molar KOH mix, and then transfer that into a strip tube. We'll neutralize that sample, and then we're going to carry that on to do a qPCR reaction. And what this qPCR reaction is doing is determining the appropriate amount of SI-PCR cycles that we need to carry out to effectively amplify our sample for sequencing. So we're going to carry out a fast qPCR reaction, and then we're going to determine the 25% peak of the RFU value for these curves. So we determined our CQ value, which in this example, 25% of the peak value comes out to be 14 cycles. So for this particular sample, we would amplify, we would do the SI-PCR step where we're adding on our sample indices, carrying that out for 14 cycles. So that's our final step. We carry out that sample index PCR step. This is going to apply our I5 and I7 dual indices to these final libraries. And we are going to amplify our library to a sufficient amount so that we can apply that towards sequencing. So this is going to be our final library that we're going to generate. So we have our P5 and P7 sequences for sequencing, and then we have our ligated probe insert in the center of this library. This insert is going to be about 50 base pairs in length, and so we'll sequence that insert for 50 base pairs. It also, though, is compatible if you have other dual indexed 10x libraries. You can also pool those libraries together and just increase the length of that transcript read beyond 50 base pairs if you desire, and you can, it's still then compatible to pool those samples together. The final thing to consider then is the amount of sequencing depth that we're going to utilize. So Rosha had alluded to this. The amount of sequencing that we're going to use is 25,000 read pairs per tissue covered spot. So when we're determining the amount of sequencing that we need to have for each one of our samples, what we're going to look at is calculate the rough percentage of the number of spots that are covered. This is something that we can do using our loop browser, so we can trace over our tissue and determine the number of spots. You could also use something like ImageJ when you look at the amount of tissue that's covering each one of your capture areas. We'll determine that percentage, and then we can calculate the required minimum amount of sequencing that we need for each one of our samples. So in this example, when we have 60% coverage, we're just going to multiply 0.6 times 5,000 of those total spots on our capture area times 25,000. And so for this sample, we would need 75 million read pairs of sequencing for that sample. So that really concludes an overview of our workflow. I just want to highlight a couple of the resources that you have available to you. On our support page, you can find all of the Demonstrator protocols as well as user guides for our Visium as well as Visium for FFPE solutions. And I will provide you the link for that support page. You also can find our public data sets. So this is complete data sets that you can download and play around with. So you have all of the raw data if you want to practice processing that. You can also have the final data sets just so that you can see what kind of information you can get from these FFPE samples. So I just want to highlight, you can find our support page at support.10xgenomics.com. That has all of those resources that I just showed. You can also find there our Q&A knowledge base. So this goes through some common questions and answers that you might be interested in. And if you have any other questions, feel free to reach out to myself or anyone else on the team, but you can also reach out to our support team at support@10xgenomics.com. I also just want to highlight a couple of other items. These are some of our newer products that are either just recently launched or are going to be coming online in the not too distant future. So the first one is our Chromium X Instrument, and this is a new instrument for our single cell solutions. And this is allowing you to now do high throughput assays. So we can, in a single run, do an experiment that involves 1 million cells. So that's really cool and really unlocks some amazing experiments. The other thing that we have coming in our Visium lineup is the ability to do multiplexed proteins. So we can evaluate panels of proteins around the area of 50 to 75 proteins in a particular panel. And that's going to be overlaid with our gene expression data on the Visium slide. So that should be coming out in the second half of this year. Additionally, coming out next year is our Site Assist instrument. And what this is going to allow us to do is take sections that are already affixed to a slide. And essentially, we can carry out the workflow for the Visium for FFPE solution using this instrument in an automated fashion, using those sections that are already applied to a slide using some special Visium slides that we're going to develop. So with that, I just want to say thank you from the entire Northeast team. We are always here to answer any questions that you might have. And so with that, I will open the floor to any questions that might not have been answered.
Very nice. I have a question about the slide having 4 little windows.
Mm-hmm.
How does the system treat each of those 4 slices? Is it integrating all 4 sections or treating them separately? Because I saw in the data slides or the data you showed, it was usually just fixing on one tissue slice.
Right. So when we if I jump back here, when we are going to denature those probe sets, we're going to capture them and then separate them into individual tubes. So we're going to process those individually. And then when we generate our sequencing libraries, each one of those is going to get a unique sample index. And so we can pool all of those together on the sequencer, but then demultiplex those individually.
I see. So each window is unique and independent. And
so each window, yes, each window would be an independent sample that you would treat as a separate sample. So up until we actually denature those, all of that tissue, it's going to be in place. All of the transcripts are in place until we actually denature the probes from our capture oligos on the gene expression slide.
And that 100 million read depth requirement would apply to analyzing all 4 of those windows simultaneously, or 100 million per window?
So it's 25,000 read pairs per tissue covered spot. So remember, each one of those capture areas has 5,000 spots. So that's where you would have to evaluate how much. You may not have the same amount of tissue covering each one of those spots on a capture area. So each one might be a little bit different in the amount of sequencing reads that are necessary.
Okay. Great, thank you. Anyone else have questions you can ask or unmute yourself?
So there's a good question in the chat. Does coverage change greatly depending on the time of fixation? I would need to fix samples overnight.
So sample fixation, that's a discussion that you're going to have with either Shoni, our tissue specialist, who has a little bit more experience with some of the fixation methods. That is something that you will have to consider for each sample, depending on the size of it. We don't give specific recommendations on fixation and embedding, simply because there's such a large variety in the field. So that's something we can probably have a follow-up conversation on, but I don't have specific guidance on fixation methods.
it wasn't to do with methods. It was more about, if you use formalin for 3 hours versus 16 hours, is there a great difference in outcome?
Ultimately you would have to test that on the sample, and it's really that DV200 score that we would evaluate to see whether there would be an impact or whether that would be okay for the assay.
Right. Okay.
I think there was another good question earlier about with Visium spatial gene expression being an available method, do we still need to do single cell sequencing? Roche, Stephen, I don't know if you guys have some examples of folks that are trying to integrate Visium data with single cell or nuclei sequencing. I know, Roche, you kind of touched on it on your data explanation during your slides.
Yeah, I can go first. So I would say you don't have to, but a lot of people do. And the reason being, some of the examples where I've seen you don't have to do single cell sequencing is you're happy with the resolution of the data set you get. So usually on each of those spots, we'll say you'll get about 1 to 10 cells in that spot. And that's depending on how tightly your cells are sticking to each other or how closely they're bound to each other. But you'll also remember there is a little bit of space in between those spots. So there's also some dead space there as well. So there are some publications that are clearly just Visium data set. The tissue is large enough that the kind of conclusions that are drawing from the data set, the resolution is fine. But there are some publications where there's multiple different nuclei that seem to be in the same spot. So they kind of want to go deeper into that data set. In those cases, people will take part of that tissue and do single cell, because with the single cell data, you can guide the cell annotation on the Visium spot. So even within the single spot, you can do subclustering. So if you really need that single cell resolution for your particular tissue type, again, this is going to be dependent on the tissue type and what the context of the cells are in the tissue. It's an option to do single cell along with the Visium example as well, experiment as well.
There are questions regarding the logistics of how much the experiments cost, and whom to address the questions. That would be Yuri from our Yuri Alexeyev from the core facility, single cell core facility. He's not here right now, but you can go on the web page of BU microarray and sequencing facility, and you can find information there. And Yuri, when he's back, will be the best person to address those questions. Any more questions?
I just had a quick question. This is maybe, of course, can be addressed by Yuri probably more specifically, but just in general for the 10x team, is at what point is a good stop for the user? Like, for example, I can imagine the user might do the H&E component and do the imaging, and then kind of hybridization might be done, or kind of subsequent steps by the core. Is there just a standard, just across the board place where a user might do some part, just like they would do the dissociation maybe for a sample, and then pass off that dissociated sample for loading onto the 10x instrument?
Yeah. With the FFPE solution, there's a number of safe stopping points throughout the protocol. So generally, I would imagine right after imaging would probably be the most appropriate stop point, because there you can pause it after you do that H&E imaging prior to doing that probe hybridization step. That's probably the most natural break point where you would want to transfer that over to the core to do the processing. Because of the probe hybridization, that step's going to be taken out overnight, and then you're going to proceed with the rest of generating the library steps. So that if that's going to be done by the core, that's probably the most likely place that you'd want to hand that off.
Thanks. And then maybe one added question is, does that step between the imaging and kind of need to move to the library hybridization, does that need to happen right away, or can you kind of stop there for more than a day or overnight?
Right. So after you have imaged your sample, you'll remove the cover slip from that. You can dry the tissue and then place it at 4 degrees. You can either use a slide mailer, or you can even just put it into a 50 mil conical. It's good to apply a desiccant packet in there just to make sure that it is not getting a lot of moisture. But I believe the stopping point is 2 weeks at that point. I might be wrong on that, but if you look in our user guide, and I can follow up with you, there is guidance as far as how long you can store those samples between the next point.
Great, thank you.
I just had a quick question. There was one question that got a little bit lost in the chat about the compatibility with the Zeiss Axioscan or the Vectra Polaris imaging output formats. I believe those are QP-TIFFs, so it's like slightly different. Is there compatibility with that? And also, is there compatibility with TSA multiplexing, which does require multiple rounds of antigen retrieval, so treatment with different like pH buffers as well as like microwaving and things like that?
As far as the imaging specifications, it's TIFF or PNG files are going to be the preferred input into the Space Ranger pipeline. There is a lot of variability in those, so that's something that usually, as long as you have the raw file and then can convert it into the TIFF, that should be acceptable. As far as the second question to using something like a Vectra Polaris, where you're doing those subsequent rounds, I am not aware of anyone that has done it yet. Theoretically, it should be feasible, but what is not known is whether that would have any negative impact on the transcripts, because that's going to be done beforehand. So it theoretically is possible, but it's going to be an experimental approach to do that. So if that's something you're interested in, I'd love to have a conversation about that.
Yeah, I'll send you an email.
Okay.
Thank you.
Yep.
I have another question about the bioinformatics that will be required to analyze this. Is the bioinformatics going to be more involved than the standard single cell RNA-seq with the Cell Ranger and the C-Loop browser? Or
it's very highly analogous. So with Cell Ranger, you're simply inputting the FASTQ files, and then you're going to get the output expression matrices. With Visium, you need to input the FASTQ files, but you're also going to provide the image file, and then you're going to do an alignment. So the software tries to do a manual alignment so that you overlay and match up that fiducial frame. So that fiducial frame is how we connect our imaging data with the gene expression data. It'll try to do that automatically. If it is not able to do that, there's a way that you can do that manually using the Loop browser, and you just choose the dots. And then once you have that orientation, it's able to then process everything through an algorithm that's very much similar to what we do in Cell Ranger Count. So the output of that is that you get your C-Loop file that you can visualize your cluster data, your t-SNE plots, your UMAP plots, as well as overlay that over the tissue. So you can see the expression overlaying on the Loop browser. You'll also have your gene expression matrices. So if you wanted to, you could take that into Seurat or other third-party tools and do further analysis with that. But as far as the computational end of things with Space Ranger, it really is highly analogous to Cell Ranger.
Okay.
Stephen, I have a question. Could you speak to the protein multiplexing workflow a little bit?
I have very little information myself on that, so there's not a whole lot that I can provide. They are going to be pre-designed panels initially, is my understanding, and most likely that will be human and mouse to begin with. But common, immunology or cancer proteins of interest, and I think over time that will then be enabled that we can do additional proteins or customization to those panels. My understanding, it's going to be in the range of 50 to 70 proteins that we can profile at one time, which compared to what you can do with immunofluorescence, or even if you were doing something like a Koya bioscience, you're still at a much higher plexy there. So all of that will then be read out as an NGS readout. So it's very similar to using our feature barcoding approach, where you look at antibody barcoded antibodies on the surface protein of cells and then pass that through our 3' or 5' single cell approaches. Here, what we're doing is capturing those proteins in the tissue and then profiling them with the Visium readout. My understanding is that's first going to be FFPE enabled, and then will also then be fresh frozen. But some of those details may change when things ship.
Okay, great. Thank you.
Mhm. Are there any other questions?
If there are no more questions, I would like to thank our presenters for wonderful information about this very cutting edge technique. And please join me in thanking the speakers.
Thank you.
Thank you for your time. Actually, there was one question about a way to get this presentation. Ala and Kim, is it best if we send it perhaps to you and you can distribute to the group?
Yes.
Okay.
We can upload it on the page. Yeah.
Great.
Awesome.
Okay. Thank you very much.
Thank you.
Thanks, everyone.
Thank you
For having us. Bye.